based models in drug discovery. That means it is an interdisciplinary field where artificial intelligence meets the biology. So large language model is actually trained on text data to understand the and to generate the like to understand the English language, to process it, to understand the context and in the English language, and try to translate the English language or to give an answer. Nowadays, we are using the chat GPT. Even in our WhatsApp, there is Meta AI. Sometimes if we ask some question to Meta AI, it gives us a answer. So this is just a large language model, the Meta AI. So chat GPT, like that, a large language model for answering the question. We usually use the Google Translate, which is actually the large language modeling for translation. So there's many tools nowadays available which handle the text data to understand the human-like language. Case of this are language model. This is just a probabilistic model. What the large language model tried, it trying to understand or capture the probability of the next word from a given sequence of words. If someone is giving the Sang is a city in, what would be the next word? While giving that sequence of word, it tried to predict the probability of the next word and which word has a maximum probability or likelihood. It give me us as that is the answer. So this is just a probabilistic model and is trained on a neural network to predict that probability. Basic building block of the any language model in the transformer era is a transformer. So transformer is also has encoder and decoder. Mainly the base of the encoder and decoder, nothing but the neural networks. So if we discuss about, tell about some popular LLM nowadays, in 2018, the BERT and GPT-1 came. And then GPT-1 improved to GPT-2, 3. Initially, it was handling the text. Now, in the GPT-4, it is published in 2023. It can handle the multimodal data, like it can handle the image also. Now the DeepSeek came from the China, and it's actually less computational unit than the ChatGPT. So it become a famous one. So now, but the basics LLM is a BART and GPT. So what is bidirectional? The full form of BART is bidirectional encoder representation from transformer. It invented or developed by the Google, and it actually captured the bidirectional context. Like if I give a sentence, the man went to the market to buy fill in the blank of milk. How the bird will fill that blank, it will understand the sentence context from the both sides. And it will give a probability to all of the words in the English vocabulary. And then which is the probability, it will fill in the blank with help of that word. So that bidirectional word, the bidirectional representation for transformer is very popular in biology. Because in biology, we just want to know in an amino acid sequence, what is the probability of one amino acid from the both sides. Apart from that is GPT, generative pre-trained transformer, is developed by the OpenAI, but it is not bidirectional like BERT. It is unidirectional. So it uses the autoregressive model. What do you mean by the autoregressive model? Is just understand the previous sequence or the previous word and try to predict the probability of the next one. It is from the left to right. GPT is also used in our biology or drug discovery to generate a new molecule. Integrative biomolecules, I already tell, but let's give that answer later. At first, we should understand drug discovery to identify and develop a new pharmaceutical compound or therapeutic agent to prevent the disease. So why we want to do that? We want to fight with the disease. We want our long life. So, but already several drug is there in the market. There's many disease already for a disease drug is available. Maybe there are few disease where drug is not available. The not only new diseases coming like the COVID-19 drug is not there. For the cancer till date drug is not there. So why we need so though why the drug discovery is so important? Already we have addressed different diseases. Why the drug is important where which drug is already exist. Like we know whenever we are having a drug for some disease or some treatment, its drug has some side effect. That means it is toxic to our body. So as a drug discovery scientist, we want to reduce the toxicity or the side effect of the drug in our body. Therefore, we need a new drug. If that one drug is already FDA approved for a particular disease, but may its side effect is more, we want some other drug whose side effect must be less. In other side, the low cost drug. If some drug have a very cost, maybe the whole population cannot use it. We will always try to make some drug with a low cost. And think, maybe you heard about the resistance, antibiotic resistance. Sometimes you are having antibiotic regularly. What happen? Our body resists for that. Our body does not, if I have that bacteria in my body, maybe the bacteria in my body is mutated and the existing drug is not working effectively. So that multi-drug resistance or drug resistance is a global health threat for human society. Like there was, you know, about the tuberculosis, there is a virus. So there was some drug. But after a few days, the bacteria, tuberculosis bacteria is mutated. So those drug is not working. It's became multi-drug resistant tuberculosis. Now, from the multi-drug resistant tuberculosis, whenever we invented some drug for the multi-drug resistant tuberculosis, sudden again evolved and it became the extreme drug resistance tuberculosis. So that's why always we need a drug for the existing, exist for maybe some there for the existing disease, but still we need that drug for to lead to prevent from the particular disease. So in drug design, there are two phases, drug discovery and drug delivery. The drug discovery phase, as a computationalist, we can make it more faster or speed up the drug discovery. Or, but in the drug delivery, where the preclinical test, clinical test is there, we cannot optimize the time at that result. In the drug phase, there is three steps. One is target selection, then lead identification, then lead optimization. So nowadays, people are working in different area of the drug discovery, like drug synergy. What is a drug synergy? It has taken two drugs together. Then the function of one drug is boosted by presence of other drug. So like in cancer treatment, usually we use the 2 or 3 drugs to treat the cancer. So therefore, to know that how drug combine work is important. Another is drug toxicity. If one drug is more toxic to our human body or not. What drug bind to our target or not, it will inhibit or it will stop the function of the target or not. People are doing research in that. Drug repurposing. When some drug is already in the market, maybe one drug is in the market for the Parkinson disease, can we use that drug for other disease like Alzheimer disease or not? People are working in this area. Precision medicine. What is a precision medicine? Like a medicine for particular for a group of people. Though you and me sharing the same DNA, but our gene expression is quite different. Therefore, my immunity system is not similar with your immunity system. So which medicine will work in my body may not will work efficiently in your body. Therefore, we need a precision or personal medicine for a particular individual or for a particular group of people. So that's comes precision disease. Even the drug response prediction, that how much drug dose we have to take for to prevent for that disease. So people are working in different research domain in the drug discovery. So let's discuss about the fundamental biomolecules in our body, which is involved in drug discovery. Like we know in our body, cell are there. Whole of body is accumulation of the cell. Within the cell, there is chromosome. Our human body has 23 chromosomes, 23 pair of chromosomes. This chromosome, nothing but the DNA. That actually is a language of human genetic, or is a language of human behavior. How I will talk, what would be my skin color, it depends on my DNA. So from that DNA, it generated the RNA, which is called transcription. From that, it synthesized the protein, which is called translation. The mainly in our body, protein is a main functional unit. Like we are talking, we are moving our hand, our muscles are collagen protein. So in our body, the main functional unit is protein. So our main target is protein. If we want to prevent a disease, we have to at first identify which is causing that disease. That called target selection. If we are able to identify which protein is causing that disease, what is the function of that protein, then only we can proceed further to develop a drug for that particular protein. So that target identification is really important. Nowadays, people are also targeting the RNA, like there is many small molecules which is target an RNA and stop to generate some malfunction protein or some bad protein in our body. But in our discussion, we'll talk the target as a protein. And what is a drug molecules? Drug foreign body in our human foreign molecule in human body. So like when we take orally, like aspirin or paracetamol, these are a small molecule. So which we can take from the oral. There are two type of drug. One is we can take from oral. Another is peptide drug. This is little in the bigger size. We cannot take it orally. If we take it orally, there will be some consequence or from some side effect. Therefore, this kind of drug we try to take from intravenous, like insulin. If you notice some of your friends have diabetes, they always take insulin through an injection. So this is peptide drug. So there is two type of different drug. One is small drug and another is peptide drug. Even our vaccine, like mRNA vaccine. This is mRNA we are pushing into our body. We'll push through a intravenous. We cannot have it orally. So this is the target is our protein. Our protein has a function. Maybe this protein is cause the inflammation in our body. And we are taking one peptide in our body which is bind with that protein and stop the function. Like this is actually HIV protein, HIV protease. So this protein actually leads to the HIV disease. So if we have take a medicine, that small molecule will bind with that protein and it will inhibit or stop the function of that. That's where drug work in our body. So now what is protein? The protein or what is peptide? Protein and peptide both amino acid chain. Protein are long amino acid chain. Like there is one amino acid building a chain. And peptide is a short amino acid. Long bits it can have in 100 amino acid, 200 amino acid. But in case of peptide, which we take as a drug, it are very small. Maybe 5 amino acid in length or 10 amino acid in length. Different in amino acid in nature, natural amino acid. Their backbone structure are similar, but the side chain have different chemical block. So that it has a chemical block R1 and it has a chemical block R2. Both that chemical block property like hydrophobicity, charge are quite different. So chemically they are different. So there is 20 different amino acid in our body. That means 20 different chemical block. Each chemical block is by an English letter in a protein chain or a peptide chain. Like this chemical word with M, this chemical block with N. So from the computer science view, there are 20 different letters. Like in our English language, there is 26 different letters. In our protein language, there are 20 different letters. Protein or peptide language. We can tell it amino acid language. The protein means it can have more than 100 amino acid in a chain. Peptide means it will have less than 50 amino acid or 30 amino acid in a chain. Even like how that amino acid important, like let's assume this peptide has the activity of anti-cancer activity. Maybe if you have take that peptide, it will bind with some protein, it will act as an anti-cancer. It can prevent the cancer. If I change one single letter here, its functionality can change. So one single letter change in the amino acid sequence, its functionality can change. So the function of a protein or peptide actually is encoded into a sequence. Even how a protein will take a structure, it also encoded into a sequence. The sequence is very important for a biomolecule. If you think about a small molecule, like which we take orally the drug, aspirin or paracetamol, that also can represent by a SMILES. The full form is simplified body, okay, molecular input line entry system. So one drug molecule can represent by a unique string. This is called SMILES string. So one drug molecule has a unique SMILES string. Now we know that how do LLM integrate in a biomolecules. So biomolecules, if you think about protein and the drug, protein is an amino acid language with 20 different letters. Like in our English language, the word cow and spoon is quite different with each other, right? As human, I can understand the cow and spoon are different. Now we train the large language model such that they can make a difference. In a similar way, in a amino acid language, the letter D and K chemically differ. So these are quite different with each other. So that last model of a protein or amino acid language should understand that. So actually the one letter represents one chemical unit. Like A represents the alanine. This is a chemical name of which is associated with the A letter. Language, it has some atom, B, C, N, carbon atom, hydrogen atom, and it actually has a symbol like this. One single dash represents single bond. Double dash represents a double bond. Hash represents the triple bond between two atoms. So the smile is a language which have some character as symbol, is as a token or is a vocabulary. Even not as character or symbol, sometimes it has a group of character together in the vocabulary. Like third bracket N plus third bracket represent the positive charge nitrogen. Idea that how the biomolecules can be represented as a language. Now the application as language model in drug discovery, where we can apply. Like the drug discovery has a three phase: target selection, lead identification, lead optimization. So for the target selection, the protein to predict the protein druggability. What do you mean by predict the protein druggability? To predict the protein function, does that protein has a binding pocket or not? Is that protein is physiologically relevant for that disease or not? Can we predict if someone give me a sequence, or can we predict if someone give me two protein sequence that these two protein sequence will interact or not? For a particular disease, particular protein doesn't, only the particular protein is not associated. If you have an Alzheimer disease, six target protein are together is acting for a Alzheimer disease. So you have to understand between protein also. So now if I want to form that research question as a LLM, so what will be my input? My input will be the amino acid language from that protein. Can I use the LLM to answer the question? So this is about the target selection. In lead identification, why I am telling the lead? Why I am not telling it a drug? When only FDA approves a molecule as a drug, then we can tell that as a drug. Before that, technically, we should tell it as a lead, or we should tell it as a drug-like molecule. So how the lead identification research questions are, like if we definitely want to know if a ligand bind with a protein. So at the first question, ligand will bind or not. If it's bind, what would be the binding affinity? How efficiently it is bind with a target protein. If it's bind a protein, is it particularly binding that protein? Because in human body, there are thousands protein are there. When we're having a drug for a particular target, may it's bind with other protein also. Then what is the side effect? I should produce a molecule which will be selectively by target. It should not be affect other protein in a thousand protein. So you should know the selectivity of that particular ligand. If a ligand bind with a protein, how its effect is inhibition of the function that can be predicted by the IC50. So that ligand protein ligand interaction we explore to identify a lead molecule. And also can we generate a lead molecule for a specific target or specific multi-target which not exist in anywhere. I want to generate computationally. Like like in the lab, people synthesize a molecule. Can I synthesize a molecule from of a computer? That question also can be addressed by the LLM. So in that question, we need two sequence input. One is amino acid, another is unsmile sequence, and we can apply the LLM to give an answer. Phase of the drug discovery, the lead optimization. Okay, I have identified a lead. It is bind properly with a protein. It its actual IC50 is also efficient. So now the question, may this drug has some side effect in our body or it's toxic to our human body? Can you reduce the toxicity? Or if I generate a drug in computational, is it synthesizable in a lab or not? So I should generate a drug which is synthesizable. Or if I have a drug with maybe I need maybe 500 nanomolar for inhibition. Can I reduce that like 1000 nanomolar? If I can reduce the drug inhibit the concentration in our body, that lead less toxicity of our body. So that's called lead optimization. Generate a drug with a new property or with a new function, that also come under the lead optimization. So how can we help of the LLM? If we use a sequence and then the LLM, it can use the sequence will be input. Then if we use a LLM, can it help us to give an answer? So the protein language model. In the first we discussed there is so many language model to handle the English language. Now in 2020, first protein language models come is PortBART. It's actually a BART model which is trained on protein database. What is protein database? There is a UniProt database which is safe or will have refer all sequence of the protein. So all the BART protein model pre-trained on that database. In 2000, AlphaFold 2 came, which identify the structure of the protein. AlphaFold is also large language model, but it doesn't only take the protein sequence as the input, so it is little complicated. It takes other input also to predict the structure. Then ESM2, another large language model, which take the evolutionary information also. 2023 protein gene came, which helped to generate or design a new protein. It helped in protein engineering. So there are several protein large language model is already there. People are working on that. So we'll know details about the port part. So port, as I told, the port BERT is based on the BERT model. So BERT language. So or BERT architecture. So what is a BERT architecture? It takes input embedding, and then it has a stack of encoder, and then it's processing. So what is input embedding in case of the protein? Definitely the amino acid. So the input sequence is the amino acid. Now we tokenize. Like in English language, we tokenize in a word. We piece a sentence in a word. But in a protein language, we piece a sentence in amino acid sequence in a letter. So it is an amino acid token. So at first, the sequence will be tokenized into amino acid. Each amino acid will give a number, numeric number. This number comes from the vocabulary position. So we have a 20 in our protein part, the vocabulary only the 20 letter. Is a very small vocabulary with respect to the English language. English language, the main vocabulary are the word. Now, each letter has one numeric word. Then this one numeric value is mapped to a vector in protein part, which is called token embedding. This vector, so one amino acid is mapped to a vector of length 1024. So what this vector represents, the token embedding, is try to capture the proper physicochemical property of an amino acid and also the evolutionary relation or evolutionary property of an amino acid. So how that if that 1024D vector, if we can map into a 2D dimension, we can notice that which amino acid have a similar property, it will come, the vector is close to each other, it will come in same direction. Their cosine angle or cosine similarity will be, the cosine angle will be less and cosine similarity will be more. And if two amino acids different, then it will be far away from each other, these two vectors. Like you, who are from the biological background, we know that K and R, like lysyl and arginine positive charge, and aspartic acid as negative charge. So if we can map that 120 dimension vector into a 2-dimension vector, we'll able to capture that. Now we have a token embedding. But in token embedding, we are capturing the property of the amino acid, but we are not capturing the sequence property. If we have two sequences like KLM and MLK, these are not same. Their functionality will be different in a practical. So you have to capture the order of the sequence or order of the amino acid in a protein sequence. So that is done by the position embedding. So position embedding is another vector of 1024D, which capture the position of a amino acid in a given sequence. So now the position and embedding and the input embedding is we concatenated together, and it's become an input embedding of a next layer. So what is the next layer in the BERT? Before, this is a stack of encoder. So what is an encoder? Encoder has a two unit. One is the attention unit, multi-head attention. Another is feed-forward unit. So the encoder has a two unit or two layer inside it. One is the attention layer, another is feed-forward layer. So whenever it is the series of encoder, that means it has attention and the feed-forward network, it has another attention network, attention feed forward network. What is the self-attention layer? What is the association of multi-head and the self-attention? Multi has nothing but a parallel self-attention layer. Okay. So there are several self-attention layers work together. Here too. I am not going into a depth, but conceptually means I am not going into the mathematics depth. But in conceptually, like in positional embedding, we capture the order of the sequence. In token embedding, we are captured the property of amino acid. But we are not captured that in a sequence, maybe it is how that first amino acid give a importance to the third or other amino acid in a sequence. When we are capturing the position of the embedding, we are capturing the sequence order. We are not capturing the context, that how the relation or how of the amino acid with all amino acids in that protein sequence. That is captured by the attention layer. So after the attention layer, in the attention layer, there is an input, is called the input. And from the attention layer, we are getting another output, which is the same embedding, but is called contextual embedding. In that contextual embedding, we are taking care that how one amino acid give importance to other amino acid. Right? When we are reading a paper, we doesn't give a important each and every letter. So we give maybe important in some key letter. So we are giving in whenever we are going to a class also, we don't give attention to each and every student. If we give as attention from some student. So they have some weightage for us. In the same, we give some attention into some word or some amino acid, we get more weightage for that sequence. That is captured by the contextual embedding. Now, this embedding is went to the feed-forward network. Network learn that embedding proper and efficiently, and that embedding is become the input of the next encoder layer. In that way, in a stack of encoder, like as I told that a bird is a stack of encoder, we're learning that embedding feature for a amino acid. How we're learning, we are trying to improve the embedding feature such that it can capture the functionality and all aspect, right? The protein physiochemical property, evolution property, structure, and also the association of one amino acid with another amino acid effectively. That's why we need a lot of encoder in a stack. Now, in the output, what we are getting, if I am for each amino acid, actually, embedding of 1024D, and for I give a protein length L, it will have a matrix. So this is the output embedding from the encoder layer. So when we are pre-trained in a large language model, there is two phases. One is pre-trained phase, another is fine-tuned phase. Pre-trained phase. Okay, let's tell what the pre-trained phase. So what's the pre-trained phase? In the pre-trained phase, we use a large database to understand the database for the processing. Okay, but how do we process in pre-trained phase? We try to, there is a term, is called masked language modeling. Whenever we pre-training, why we're pre-training our database? To learn that embedding, the token embedding, what would be the positional embedding. We learn in the pre-trained model, we use the masked language modeling. What is the masked language modeling? We mark some amino acid in the sequence and predict what would be the amino acid. Then if we predict properly, we know that is our starting positional embedding, and this is our pre-trained model parameter. What is actually parameter? Machine learning. Machine learning, what we do, we have an input variable, where input variable is here, maybe the sequence, and we have to map variable into the output. Let's assume it's a function of a protein. But to mimic that, to identify function, we have to take a function. And in that function parameter, we cannot change the input. Input is sequence. What we can update the parameter. So in the pre-trained model, we learn the parameters. Parameter of the attention layer, parameters of how we'll represent the token embedding, starting token embedding, parameter of the starting positional embedding. We learn the parameter. And how we learn it? We learn with help of the mass language modeling technique. Usually in Protbird, 15% token of an amino acid, we make a mask. That means we don't tell that model and we try to predict that model. And this way, we make our out. We fixed our embedding parameter. We fixed our model parameters. Now, fine tuning. We have already fixed our model parameter with a large data, which can capture properly. So in case of protein, we pretend to capture properly the protein amino acid property, evaluation information, and like structural property. So you are happy with that. Okay, our pretend model can capture all of this property. But does it capture particular function? Like, is it captured that this peptide or these molecules has a cancer activity or not? May the pretend model properly, because it is dealing with a large data set, not a particular task-oriented data set. So we need a fine tune. For that, for a specific task, we need a small data set. But for the small data set, if we start from the crash, lead in an error. Why? Because whenever you start with small data set, it may not able to learn the proper physicochemical property or the evolution property of the amino acid. So that's why we need a pretend model. Whenever we have a pretend model, then for a particular task, we can take that parameter and we can make our fine-tuned model for a particular task which is associated with a small data set. This is a fine-tuned of a pretend model. What we else can do, as I tell that this is processing. In the output, actually, if we do a fine-tuned model also, in an output from the encoder, I can get a embedding, protein embedding. That embedding, instead of the fine-tuning, I can use that as a feature input of my some other deep learning architecture to predict some other function or to improve the performance of the model. So, like a similarity, like we have a protein large language model, and the same we have a pre-trained large language model for the chemical, for the SMILE, like ChemBird or MolBird, like molecular graph bird in 2024. So these are the chemical large language model. But in a ChemBird also, what way it has the embedding, input embedding, tokenizations, then its positional embedding is take the input embedding, then the encoder stack, then the output processing, same sequence are there. But in case of the chemical language, that embedding is a two-step. Like at first they do a character level tokenization in a chamber, but maybe the character was not important to capture the property of a chemical molecule. So the second step, they mark some frequency substructure. So like they mark some, these are, okay, this is a single bracket equal to O single bracket, but actually together is called the double bonded O. So in tokenization, there is two steps. One is at first is to tokenize as a each character, then it's marks based of the substructure of a model. Of yeah. So this is a tokenization. After the input tokenization is to the encode positional tokenization, then encoder, then the output process. Now the application of the protocol, where people applied. In 2023, there is a paper published in Protein Science. This is a AMP bird. What is the full form of AMP? Antimicrobial peptide. So they have a peptide data set. It's very small. 1,778 AMP and 1,778 is non-AMP. And they use the pre-trained BART model to find the protein embedding feature instead of physiochemical feature, evolution feature differently. They are taking the protein embedding feature which already captured the all chemical property, evolution property, structure property. So that sequence and 1024D, this feature they set into the fully connected layer and then a sigmoid node to identify it is has a antimicrobial peptide or non-antimicrobial peptide. Data set we cannot pretend. We cannot capture the physiochemical property. So that's why they did the fine tune. They take a small data set and start with the pretend model, which already capture the physiochemical property. And then now they use it to identify if I give sequence, it will tell it has an antimicrobial peptide or not. And they compare with the existence paper that their BART model is far more better with respect to the other existing normal model. In 2021 into the bioinformatics, they actually work with also a fine-tuned model. What they use a protein BART. They have a sequence, they have a data set like some peptide, which is a test, and some peptide doesn't have a beta test. Why it's important? Like if you notice, if you give a medicine to a child, they don't want to have it. Even we adults also sometimes. So if we can make a medicine without beta test, may it will help to handle that kind of situation. So to address that one, they have made a prediction model that if you give a peptide, that model will predict it will have a beta test or not. Processing also. Like they has an input as sequence with help of the protein part. They have a amino acid, they have a vector. This vector for each amino acid, they use an input vector in their bio-LSTM model. So this is not only fine-tuned. They use the output embedding into some other deep learning model to predict the function. They have also shown there, but embedding actually more better than the other machine learning model. The next one is graph BERT. Graph BERT is actually architecture, but like in the BERT architecture, there is token embedding and the positional embedding. But in graph BERT, what they have, graph is the connection between different nodes. So graph has a node embedding and the graph embedding and the structure of the graph. What is a node embedding? Like who know about the graph neural network? In the node, we use a feature vector to which we call the node feature. So this actually, each node is represented by a node embedding in a graph. Then we add the positional embedding. That node is connected with other node in that graph. This is called the positional embedding in the graph neural network, graph BERT. And what is the structure? They try to understand the structure of the BERT with help of the degree and other property of the graph. So these all add together in an embedding input embedding layer. Then we have an encoder and the output embedding. So what in this paper they did, they use the graph embedding and the make a framework for the protein-protein interaction identifier. Like if I give you two amino sequence, will it interact or not? That protein you will interact in or not, that will identify. What is required? It is required to understand the disease. So what they did, they took a two amino acid sequence and they use a character CNN and by LSTM. So this is not a large language model. This is a sequence-based language model. So they use that model to prepare the protein embedding. These two protein embedding from both protein, they merge together. And this protein embedding is used as a node embedding in that graph. So this is a protein-protein interaction network where each node represents two protein will interact. The each node means two protein, which are interacting in a protein-protein interaction database. And this protein-protein interaction to protein, the node embedding is taken from that embedding. Then they use the graph embedding, graph BART model to predict that protein-protein interaction is true or false. And not only that, protein and in the molecule. Like nowadays, the language modeling spectrometry data. So in this Novo, where the de novo protein sequencing from the mass spec they have identified with help of the BART model. What is the smart spectrometry will give you some mass charge M by Z, and for that unit, actually specialist is mass spectrometry, they can identify the protein in that biological sample. If you give a biological sample, then who are expert in the smart spectrometry, they can analyze the smart spectrometry data and they can tell that they can generate the protein sequence. But there also, what in this paper they did, they take the sequence of the mass and intensity from the mass spectrometry, and then they use a sinusoidal encoder to encode it into an embedding. Then this encoder, they again use a decoder. Actually, whenever BART has only encoder. Whenever we are talking about transformer, it has two units, encoder and the decoder. Encoder encode into the embedding, output embedding, like from a sequence to a numeric embedding, decode it from the numeric encoding to a amino acid sequence. So they use a decoder to generate from the encoding to different peptide. So here they use a large language model. And also when they are different peptide, they use that peptide. This is hypothetical peptide. So now they take that all generated peptide. Again, they use a BART model to predict, is this peptide a decoy? What is actual peptide? What has a more probability to generate? So they showed that their model is good enough to identify a protein from a mass spec data. So, what is a mol BART? Is a pretend model. This is not a fine-tuned model. Okay. Mol BART is a pretend model. Is a pretend on a data set which is called Campbell data set. So they take the Campbell data set and Jing data set together. There is 4 billion smile. And how they make the input embedding. When you are thinking about the pretend model, we can think about new type of token embedding. So here they did not use the group of substructure. They take a smile, convert into the canonical Mulligan fingerprint. So one smile we can convert into the fingerprint, means one numerical vector, which will capture the property of the fingerprint. So it's take a smile and the smile is converted to a fingerprint. That fingerprint they use as a token embedding. And this they feed into the encoder and they pretend the whole model. And they have shown their model is able to identify different tasks like blood-brain barrier prediction, blood-brain barrier penetrating or not. It is HIV function is there or not. What is the toxicity? But if you look into that paper, maybe the all performance are not efficient. If we want to make a toxic molecule, we have to pretend that Mollberg model. There is another paper, is a sequence of that Mollberg paper. So in the Mollberg, they use only the molecular fingerprint. But like molecular, when we are thinking about a molecule, fixed. Is bond or rotating? Which are the single bond in a molecule? It has a rotation. It's a flexible. So whenever you think about the molecular fingerprint, we're not capturing the dynamic nature of a molecule. So what they did, rotary position embedding. They use a rotary position embedding. Their positional embedding based on the rotation of a single bond. Then they have the data, the rotation angle, phi psi angle, the rotomer. And that rotomer, they make a vector and they use that as a positional vector. So they use this as a token embedding, and the positional embedding, they use a rotary positional embedding. They merge it, pretend it, then they showed that how it's worked on different terms. So this is also a pretend model. This is another paper in 2023 published in maybe Frontier Molecular Bioscience. They want to capture in the embedding. This is also a pretend model, but they change the attention layer. How to give an attention? They give an input in an attention layer. They change the attention layer. How they change the attention layer? They give a 3D information and also 2D information. They did not change in the input embedding. They made some change in the attention embedding such that it can learn more about the 3D information and the 2D information. For the 3D information, they take the distance between the bond in the 3D space, X, Y, Z coordinate. And for the 2D information, they take the bond energy. So each bond has a different energy. They take that one. And these two, they feed into our attention layer such that attention should give on base of these two information. As I did and pretend we know, that is another application where they have actually generate the molecules with help, but from it generate the molecule with help of the decoder. So decoder is take input embedding new. This take a sequence, then it's take a sequence, and then is give an output as a sequence. So this is a decoder. What is a decoder do? If I give us some character which is related to the molecule, it will predict the in black color is a prediction. So it will predict the next character. It will generate a molecule character by character. How it is giving a probability. But whenever you generate a next character, like here if I give till C, maybe I decoder, and the next character is one single bracket. Is it a valid sequence? No. If I want single, there is another token which can identify. The token is end of sequence, EOS. The probability of that token in that position is 0. So it is giving it some reward which is 0. If it's given sequence reward 0, that means my sequence is not ending. When the end of sequence is high probability, that means it is a complete molecule. Take that the molecule is generated properly. When the molecule is generated properly,